Imagine for a moment if we could truly understand, maybe even predict, with incredible precision, how a single cell is going to react to something. Like, say, a new drug.
Hmm.
This has been a long-standing dream in biology, right? Almost mythical. Why is that understanding so critical?
Oh, it's absolutely foundational. I mean, for understanding life itself. Think about it. How do cells integrate signals? How do they actually carry out responses?
Right.
And then how do they form the complex systems that make up, well, everything? It's the bedrock. But building truly predictive in the silico,
Yeah.
computer-based models, that's been a massive, massive challenge.
Why is that? Just the complexity.
Yeah, the sheer complexity of it all and the incredible need for vast, really precise data to actually train those models. You need a lot of good data.
Okay, let's unpack this then for you, our listener. You've given us this really groundbreaking research paper describing something called Tahoe 100-dimmer.
Mm,
So our mission for this deep dive is to explore this giga-scale single-cell perturbation atlas. What exactly is it? Why is it such a monumental breakthrough? And, what surprising insights does it actually offer for understanding complex diseases like cancer and maybe even speeding up drug discovery? Let's dive in.
sounds good.
So historically, how did scientists try to figure out cell behavior and drug effects? We've probably all heard of bulk transcriptomics. What did that actually tell us? And maybe more importantly, what did it miss?
Right, bark transcriptomics. Well, it basically aggregates signals, lumps them all together from millions of cells at once.
Okay, an average.
Exactly. It's like taking an average of a huge crowd. It gave us a general idea for sure and provided some valuable insights into overall responses, no doubt about that.
But
But it completely masked the individual differences within that crowd, what biologists call heterogeneity. Cells might be in different phases of their life cycle or have different genetic backgrounds slightly. Or they might just respond uniquely, even to the exact same conditions. And this heterogeneity can lead to incredibly diverse outcomes that those bulk methods just, well, they simply couldn't resolve. It was all blurred together.
so it's kind of like trying to understand a really complex city like London or New York
Right.
just by looking at the overall traffic flow number. You miss the individual cars, the different routes people take, maybe construction detours.
That's a great analogy, yeah. You lose all the nuance, all the individual stories happening within the city.
And this is where things get really interesting with the rise of single-cell assays, single-cell RNA sequencing, or Fritz RNA-seq. How did that technology just completely change the game?
Oh, the cRNA-seq.
Yeah.
It truly transformed our ability to look at individual cells. Instead of averaging, you get a detailed molecular snapshot of each cell.
Wow.
Yeah. This profoundly expanded our understanding of how cells respond to perturbations, whether that's small molecules like drugs or genetic changes. Think of it like going from a blurry crowd photo to suddenly having individual, high-definition portraits of every single person in that crowd.
That's a huge difference. But even with these amazing advances, the paper points out that large-scale, perturbation-focused single-cell datasets, so datasets looking specifically at how cells react to drugs or other changes, remained quite limited. What were the hurdles still standing in the way?
Yeah, challenges definitely persisted. For one, there were these things called batch effects.
Batch effects.
Yeah, basically experimental variations between different batches of experiments. These technical differences can easily obscure the real biological signals you're looking for. It's like experimental noise.
Okay, makes sense.
Then there were issues with inconsistent protocols across different labs or studies. Limited coverage, maybe they only tested a few drugs or a few cell types. And often, maybe surprisingly, a low number of actual observations, like individual cell measurements, for specific cell and perturbation combinations.
So not enough data points for each specific scenario.
Exactly. Plus, many existing datasets focus maybe too narrowly on just one specific disease, leaving broader, more fundamental questions about general cellular behavior unanswered.
Right, so how did they actually pull off this monumental feat? Generating a data set that tackles these hurdles and scales up so massively, the researchers developed something really innovative called the Mosaic platform. Can you walk us through the genius behind that? How did it enable this unprecedented scale?
Yeah, the Mosaic platform is really clever. What's truly fascinating is its core concept. It cleverly exploits LEER-ASEC, that single cell tech we talked about, to capture high-resolution transcriptomic responses, so gene activity profiles to thousands of perturbations, all in parallel,
Thousands in parallel.
by using what they call cell villages.
A cell village. Okay, that sounds quaint. What does that actually mean in the lab?
Oh, yeah. Imagine a single experiment where instead of just one type of cell growing in a dish. You have a highly diverse and optimally balanced mixture, a village made up of tens, even hundreds, of distinct cell models all mixed together.
All in one pot.
Pretty much. For Tahoe 100mm, this meant mixing 50 different cancer cell lines together. They actually grew them as spheroids, these 3D clusters of cells, which mimic real tumors a bit better than cells just flat on a dish.
Okay, so a 3D village of 50 different cell lines. Then what?
After treating this mixed spheroid village with drugs, they use a really clever trick called genetic demultiplexing.
Genetic demultiplexing. Break that down.
Okay, so each cell line has its own unique genetic variations, right? Tiny differences in their DNA code. They can use these almost like a genetic fingerprint. After the experiment, they sequence each cell individually, and based on these unique genetic markers, they can computationally figure out, oh, this cell originally came from cell line A, and this one came from cell line B, and so on.
Wow. So even though they mixed maybe 50 cell lines together, treated them all at once, they can still tell exactly which cell was which afterwards using its DNA.
Precisely. It's like putting unique barcodes on every cell line before you mix them.
That sounds incredibly efficient. How does that pooling, mixing them all together, help with those batch effects you mentioned? And how does it let them scale up so massively?
Exactly. So by mixing all these different cell lines and running them through the exact same experimental process, same reagents, same machine, same time, you dramatically minimize those technical variations that would normally pop up between separate samples or different experimental runs.
Ah, because they all experience the same potential noise.
Right. It effectively cancels out or at least greatly reduces those batch effects. And because you're doing thousands of perturbations, different drugs, different doses in parallel within these pooled villages in essentially single experiments, it scales dramatically. You get way more data much more efficiently.
Okay, this is starting to make sense. It's a really smart way to do more with less, technically speaking. Which brings us to the sheer scale of Tahoe 100M itself. Give us the big numbers. Just how big is this atlas?
Okay, brace yourself. Tahoe 100M comprises over 100 million individual single-cell transcriptomes.
100 million.
Yep. Over 100 million gene expression profiles from single cells. They collected this from those 50 cancer cell lines, which were subjected to more than 1,100 different small molecule perturbations drugs, basically each tested at varied doses.
Incredible.
To put it simply, it is the largest publicly available single-cell dataset of its kind to date. The whole process involved culturing those 3D spheroids of mixed cell lines, treating them with drugs for 24 hours, then carefully breaking the spheroids down into individual single cells, then barcoding each cell's RNA using a specific high-throughput kit called the Parse GigaLab kit, sequencing it all, and finally using that SMP-based deconvolution, the genetic demultiplexing, to assign each of those 100 million cells back to its specific original cell line and the exact treatment condition it received.
That workflow is just staggering. And you mentioned quality control. Did they check if this whole complex process was actually reliable?
Oh, absolutely. They included rigorous quality control measures and biological replicates. For instance, they ran 2 experimental plates, plate 6 and plate 14, as exact biological duplicates, same cells, same drugs, same conditions, just run separately. When they compared the matching conditions between those 2 plates, they saw an incredibly high correlation, a median Pearson correlation of 0.97, which is almost perfect agreement.
Wow. Okay, so the platform, this Mosaic approach, is robust and reproducible. That's crucial.
Absolutely critical for a data set this size.
Okay, so we've covered the problem averaging, hid the details, and this really ingenious Mosaic platform solution that built this Giga-Scale atlas. Before we dive into what they found on the data, let's do a quick recap for you, our listener, on these building blocks. What are the absolute essentials about how Tahoe 100mm came to be?
Right, so the core problem was heterogeneity. Individual cells respond differently. But old methods like bulk sequencing just averaged everything out, losing that detail.
Got it. The averaging problem.
The solution was the Mosaic platform. Key ideas there, they're cell villages, mixing many cell lines, and genetic demultiplexing, using DNA fingerprints to tell them apart later. This allows massive parallel experiments and cuts down on batch effects, that experimental noise.
Cell villages and genetic unscrambling.
Okay.
And the result, Tahoe 100mm. Over 100 million single-cell profiles, the largest data set of its kind, covering 50 cancer cell lines, and over 1,100 drug perturbations. Unprecedented scale and, importantly, really high-quality data.
Fantastic summary. Now, let's get into the content of that data. What kind of cells and drugs are we actually talking about here? What does this massive data set cover biologically?
Yeah, the diversity is really key to its power. So of the 50 cell lines they started with, 47 had enough cells profiled for really deep analysis. And these weren't just random cells. They were derived from 13 different organs.
13.
Oh.
Yeah, with the majority coming from common cancer sites like the lung, bowel, pancreas, and skin. And crucially, these cell lines carried a diverse set of well-known cancer-driving mutations genes like TP53, KRAS, CDKN2A. About half the cell lines studied had alterations in these key cancer genes.
So a real mix of cancer types and genetic backgrounds. Why is that genetic diversity so important?
Because it's essential for understanding how different genetic backgrounds can influence a cell's response to drugs. Does a drug work better if a cell has mutation X versus mutation Y? This data set is designed to help answer exactly those kinds of questions.
Okay, makes sense. And what about the drugs themselves? Were they mostly experimental, or did they include drugs that are already used in the clinic?
It was a really good mix. They used 379 distinct drugs in the atlas, covering a remarkably wide range. Of those, 180 could be classified into 25 different mechanisms of action, or MOAs.
MOA, that's basically how the drug works at a molecular level, right?
Exactly. And a significant majority of the drugs, about 69%, are actually approved agents already used in patients, or at least compounds that have reached clinical trials. So very clinically relevant.
That's great. And what do these drugs target?
Collectively, these drugs target 325 different genes. And interestingly, 120 of those genes were targeted by more than one drug in the set.
Ah, so you can compare different drugs that supposedly hit the same target.
Precisely. It allows for a much deeper, more nuanced investigation of specific cellular pathways and how different compounds, even with the same target, might have subtly different effects or off-target activities.
That sounds like a goldmine for researchers. What really stood out when the scientists looked at the sheer data density? You mentioned scale before.
Yeah, the density is incredible. This data set captures 17,813 unique cell line drug conditions.
17,000 unique experiments, if anything.
Exactly. And to put that into perspective, the paper mentions there's a 31-fold increase in the number of drug-perturbed conditions compared to a previous benchmark single-cell drug screen study.
31 times more conditions tested.
Imagine giving an AI 31 times more examples to learn from for each cell type and drug combination. That's the kind of power we're talking about here. It lets the AI spot patterns that were just invisible to us before. And it also offers a 29-fold increase in the number of observations, meaning individual cells measured per condition compared to a benchmark genetic perturbation screen. So not just more breadth, but also more depth for each condition.
That's an absolutely astonishing leap forward in both breadth and depth. When they first started looking at all this data visually, did they see anything right away that confirmed the quality you mentioned? Did it look clean?
Yes, absolutely. They use some advanced visualization techniques, dimensionality reduction methods like SEVI and TSNE, essentially ways to map out this complex multidimensional data in 2D or 3D.
To see the relationships between cells.
Exactly. And what immediately jumped out was a clear separation, clear clustering of cells based on their intrinsic biological properties primarily, their genetic identity, which cell line they were, and their cell cycle phase, where they were in the growth and division cycle. They weren't clustering based on the original experimental plate they came from.
Ah, so the biology was stronger than the experimental noise, the batch effects.
Precisely. It's a crucial indicator of very minimal batch effects, confirming that the Mosaic platform really succeeded in generating exceptionally clean, high-fidelity data. The biology shines through.
That's fantastic. So the biological signal was strong and clear. With such clean, reliable data, what was the first thing they tried to understand about the drug's actual impact? How did they quantify it?
They used a metric called E-distance. Essentially, it measures how much a perturbed cell population the cells treated with the drug separated or changed transcriptionally from its untreated control population.
So a measure of how much the drug moved the cells in terms of their gene expression.
Exactly. It quantifies the magnitude of the drug's effect on the transcriptome. And consistently, they found that higher drug doses led to a larger E-distance.
Which makes perfect sense. More drug, bigger effect.
Right. It's a good sanity check, showing the system behaves as expected biologically.
Were there any particular drugs or drug classes that stood out as having surprisingly large effects, maybe being outliers?
Yes, there were. For example, drugs that mess with really fundamental cellular machinery, like protein synthesis inhibitors, specifically heringtonine and homoheringtonine, which directly stop ribosomes from making proteins, show particularly strong universal effects.
Hitting a core process hard.
Exactly. Dinaciclib, which is a potent inhibitor of CDKs, enzymes that control cell growth and division, also stood out for its very broad inhibitory effects across many cell lines. And more broadly, they saw that anti-cancer drugs designed to target common pathways in epithelial solid tumors, things like AMPK inhibitors, PI3K-AKT inhibitors, MTOR inhibitors, CDK inhibitors, generally showed larger E-distances, larger effects.
Indicating a more pronounced overall impact on the cells, as you'd hope for an anti-cancer drug.
Precisely. The dataset really starts to paint a picture of the overall shape or landscape of drug response across many contexts.
This is fascinating. Okay, let's quickly recap what the data itself reveals at this high level for you listening.
Sure, so first, the atlas is incredibly diverse. 47 cancer cell lines analyzed from 13 organs, carrying key mutations. It profiles 379 drugs covering 25 mechanisms of action, many clinically relevant.
Diversity is key.
Second, the scale advantage is huge. Over 17,800 unique cell line drug conditions tested, a massive 31-fold increase over benchmarks, providing unprecedented breadth and depth.
Scale enables new insights.
Third, the data quality is exceptional. Visualization shows minimal batch effects, meaning the biological signal is strong and clear, thanks to the Mosaic platform.
Clean data we can trust.
And finally, the overall drug impact follows logic. Higher doses cause greater transcriptomic change, measured by E-distance, and drugs hitting fundamental pathways or key cancer pathways tend to show larger effects overall.
Got it. So the foundations are solid. Now let's get into some specific, maybe more surprising examples of how these drugs interact differently depending on the cell's own context. This context dependency seems crucial.
It really is, and this dataset is perfect for exploring it. So it raises the question, how do drugs that target specific pathways behave differently depending on a cell's own genetic mutations? They focus quite a bit on the RAF pathway, which is, as many know, incredibly important in cancer development.
Right, RAS and RAF are notorious cancer genes. Can you give us a concrete example of how a drug targeting this pathway behaved differently in different cells within the atlas?
Yeah, absolutely. Take dabrafenib. This is a drug specifically designed to inhibit a mutated form of the BRAF gene called BRAV600E.
Okay, a targeted therapy.
Exactly. And what they saw in the Tahoe data was that dabrafenib had its most pronounced effect, the largest E-distance, the biggest change in gene expression only in those cell lines that actually carry that specific BRAF600E mutation.
Just as intended. What about cells without that mutation?
In stark contrast, cell lines that had a different mutation in the same pathway, like a KRAS mutation, which often acts sort of upstream of BRAF, showed minimal change in response to dabrafenib.
Ah, so it didn't work if the target wasn't there, even if the pathway was relevant?
Precisely. It aligns perfectly with our established biological understanding of how these pathways work and how targeted therapies should function. The drug only really hits hard where its specific target is driving the cancer.
That's a great validation. What about a drug that targets RAS itself, that upstream component you mentioned?
Good question. They looked at RMC-6236, which is described as a pan-RAS inhibitor, meaning it's designed to inhibit various forms of the RAS protein, including mutated KRAS.
So it should work in KRAS mutated cells.
And indeed, it showed its greatest effect, its largest E-distance, in the cell lines that carried KRAS mutations. This really underscores its KRAS-specific action in those contexts.
Wow. These examples are powerful because they really highlight the incredibly precise, context-dependent insights you can get from this kind of atlas. It's not just if a drug works, but where and why it works, down to the specific mutation.
That's the essence of it.
Yeah.
This is the kind of data that really fuels personalized medicine. Tahoe 100M provides that incredibly detailed map of context dependency.
Okay, so we see these specific gene expression changes linked to mutations. Did they look beyond just the transcriptome at other fundamental cellular processes, like how drugs affect the cell cycle, how cells grow and divide?
Yes, they did. That's another fascinating layer. They investigated the drug impact on the cell cycle phase distribution. So looking at what proportion of cells are in G1 phase, growth, S phase, DNA replication, or G2M phase, preparation for an actual cell division.
And did they see patterns there?
Oh yes. Distinct patterns emerged. For instance, among the CDK inhibitors, those drugs that target the cell cycle engines, they saw differences. Palbociclib, which targets CDK4 and 6, tended to cause cells to pile up in the G1 phase, stopping them before DNA replication.
Okay.
Whereas dinaciclib, which inhibits a broader set of CDKs, including CDK1 and 2, tended to enrich cells in the G2M phase, arresting them later, just before or during division.
Ah, so even within the CDK inhibitor class, the specific target leads to a different cell cycle effect.
Exactly. It's due to their specific molecular targets. Palbociclib hits CDK46, which regulates the G1 to S transition, while dinaciclib's targets, like CDK1, are crucial for inducing G2M arrest. The data reflects this known biology beautifully, but across many cell lines simultaneously.
That's such a specific, nuanced insight, revealed at scale. What about other drug classes? Did they see similar patterns or maybe
They
exceptions?
did. For example, HDAC inhibitors and microtubule inhibitors, two other classes of anti-cancer drugs, generally induced arrest in the G2M phase. That was the trend.
Okay.
However, there were interesting exceptions even within these groups. For example, carbamazepine, which is mainly an anti-epileptic drug but has some modest HDAC inhibitory activity, showed very little G2M enrichment.
So its main effect probably isn't via HDAC inhibition in this context.
Likely not strongly, no. And another HDAC inhibitor, tacitustat, actually showed a negative G2M enrichment, fewer cells in G2M. Instead, it seemed to cause arrest earlier in G0, G1, possibly by downregulating CDK4. And even among microtubule inhibitors like paclitaxel, Taxol, which you'd expect to cause strong G2M arrest, the effect size was sometimes minimal in certain contexts in this dataset.
So the general rules apply, but the exceptions and the context really matter. This data set lets you see all that fine detail.
Exactly. It captures that complexity and nuance across many drugs and cell lines.
Okay, stepping back again, what does all this mean for you, the listener, or for the broader scientific community? How does having access to Tahoe 100 MEMP actually accelerate progress in understanding biology and maybe treating diseases?
Right. The bigger picture. Tahoe 100 MEM isn't just a massive data collection. It's specifically designed, really, to power the next generation of AI-driven models of the cell.
AI models. How so?
Think about how large, diverse data sets completely revolutionize other fields like natural language processing, how chatbots work now, or image recognition.
Yeah, the leap has been incredible.
It's because they finally had enough high-quality data to train complex AI models. Tahoe 100M aims to do the same for cell biology. It provides the incredibly diverse and deep training data that AI needs to learn the complex rules of gene regulation, how pathways interact, and all those context-dependent functions we've been talking about with unprecedented accuracy.
So it's like giving an AI a giant detailed encyclopedia, or maybe more like a flight simulator for how cells respond to almost anything, enabling it to predict outcomes much better than before.
That's a good way to think about it. A simulator, or a master encyclopedia. It allows AI to move beyond simple correlations and start learning the deeper mechanistic rules governing cell behavior under perturbation.
And this sounds like it could directly impact how we approach and treat complex diseases, especially cancer.
Exactly. Tahoe 100 MEM vividly exposes the profound heterogeneity, the differences in how cells respond to drugs, even within the same tumor type or cell line. This means AI, trained on this kind of data, can help us understand several key things much better.
Okay, like what?
First, subpopulation dynamics. Even within what we think of as a genetically identical cell line, you often have different subsets of cells that might respond differently. Maybe some are resistant, some are sensitive. AI can help identify and track these subpopulations.
Find the cells that might survive treatment.
Right. Second, pathway-specific effects. It can help pinpoint which cells might evade treatment by using alternative or compensatory pathways. And it can illuminate where complex crosstalk pathways influencing each other might complicate treatment strategies.
Untangling that complex network.
Exactly. And third, predictive biomarkers. Because the atlas covers such a broad panel of cell lines with known genetic backgrounds and diverse drug responses, AI can systematically search for transcriptional patterns, specific gene expression signatures that are strongly linked to whether a cell line, and potentially a patient's tumor, will respond to a particular drug.
Finding signals that predict success or failure.
Precisely. These patterns could become powerful predictive biomarkers to guide personalized medicine tailoring treatments much more accurately to a patient's unique molecular profile.
This really feels like it's moving us away from a one-size-fits-all approach towards truly precision oncology, driven by this deep single-cell understanding.
That's the goal, absolutely.
So what we've explored today with Tahoe 100M is, well, it feels like a truly monumental step in systems biology. It's not just about collecting more data, is it? It's about collecting the right kind of data, single cell, perturbation-focused, at an absolutely unprecedented scale, using really clever innovations like the Mosaic platform.
That's right. The innovation in generating the data is just as important as the data itself.
And this allows us to finally move beyond beyond those blurry averages and truly start to understand the individual, nuanced cellular responses to a vast array of perturbations.
Hmm. This resource being publicly available is really poised to catalyze a whole new era of data-driven discovery in biology. By understanding gene function, regulatory networks, and how they're modulated by drugs and other cues at this incredible level of detail, we're building a much stronger foundation for truly predictive mechanistic models of the cell.
And the downstream effect, the hope, is that this leads to more effective, more personalized treatments for patients, especially in horribly complex diseases like cancer, where every single cell's response can genuinely matter.
That's the ultimate aim.
This whole discussion raises a really important question, maybe for you, our listener, to think about. As AI models become increasingly sophisticated, powered by massive detailed data sets like Tahoe 100 EMMA, what ethical considerations or perhaps entirely new applications beyond even what we've discussed today do you think will become most critical in this evolving field of personalized medicine? Something to definitely mull over until our next deep dive.